This is a competition that we now hold annually for images related to research. And we ask scientists to send us their most evocative images. This was from Dr. Gabor Folds and others at Imperial College. And actually, these are new heart muscle cells that have been grown from stem cells.
We are having human embryonic stem cells is a very special, exciting cell type which can give rise to any kind of cell in the human body. And our main interest is growing heart tissue, growing heart muscle, and trying to understand how we can make it properly and how we can use these cells after all in kind of situations where the heart is injured and needs replacement of the dead part of the tissue.
And what's it like being part of this competition?
Really great honour, really happy to be here, and it's one of the highlights of the 3 years I've spent in the Imperial College.
This rather beautiful image is the second runner-up, and this comes from Dr. Patrick Hales at the University of Oxford. This is using very specialised technology to look at the individual heart fibres and showing how beautifully how they wrap around the heart and explain why the heart is such a good pump. It's much better than anything else man's been able to devise.
The reason we're doing it is we try to build 3D computer models of individual hearts as accurately as possible. And one of the most important things to build these models accurately is to know the orientation of your fibres in the heart, because that will affect the mechanics and the electrical propagation through the myocardium. So by having a good representation of the structure arrangement of these myofibres, you can do much more accurate modelling on your 3D computer models.
You might think these are heart cells. Actually, they're not. These are blood vessel cells, what they call pericytes, that are growing in a tissue culture dish, and I suspect they were just intelligent enough to collect around this particular shape to give him the image that he needed to get first prize. So if you'd like to come and accept your prize.
So these cells are pericytes. They're specialized cells of vessel walls, and when you have a heart attack or an inflammation, leukocyte or white blood cell have to go through this vessel wall and cross this type of cells. And this particular experiment where we were looking at how they regulate another protein involved in the process of migration of white blood cells, because the migration of white blood cells will damage even more the tissues. So we, by this kind of experiment, we can try to manipulate the reactions and also trying to find therapeutics against inflammation.
First runner-up is Dr. Pauline Bennett from King's College London, and what this is showing is these are two heart cells, and heart cells have to communicate if they're going to beat. There's no point in having your heart cells all going off at their own free will. They've got to communicate and beat synchronously, and the way they communicate is by sending molecules across these junctions here.
In the electron microscope, we took lots and lots of pictures of a particular section through a heart, taken at lots of different angles. And in the computer, we can put those all together to form these little steps in through the structure. So this, you can see in depth what's happening through the structure in the heart.
University of Leicester, Dr. Emma Chung. What you see here are little flecks. I think you can see these little red flecks coming through. They're only coming from one side. They're running up this side here and running around these blood vessels and going up here. They're not coming up this side. And this is simulating somebody who's got atherosclerosis, narrowing of the artery lower down, and they're throwing off what we call microemboli. And these microemboli are little pieces of cellular material which go into the circulation, run up into the brain, eventually meet a blood cell that's a vessel that's as small as they are, and they stop.
Our aim is to understand the trajectories of emboli going through the cerebral arteries so that we can model them using computer simulations. And ideally, we'd like to create a virtual patient for predicting the impact of emboli during cardiac surgery, because cardiac surgery, although the safety's improved a lot over the last 50 years, there's still a small risk of stroke there.
This is magnetic resonance again, so this is real live patient, and what it's showing you is blood running through the two sides of the heart. So blue is the deoxygenated blood coming from the body to the right ventricle and out to the lungs. Red is the blood coming from the lungs into the left ventricle and out through this big vessel here, the aorta. And so the winner is Dr. Philip Kilner from Imperial College.
My video bloodstream to the heart really shows the patterns and direction changes of flow through the whole heart and aorta. It's done by non-invasive magnetic resonance imaging in a healthy young man. And what we've done is acquire data that encodes all three directions of flow in a three-dimensional data set and through the whole heart cycle, repeated many times. So we can play that back as a single movie of the heart.
Yeah, I think they're outstanding. It's our opportunity to bring a bit of lightheartedness actually to the science, because science is a fairly serious business. But by capturing these images, which also bring together the biology and the technology that we have to bring together to do really good science, I think it's a very good way of raising the profile of the scientists.